Okay, girls, today we will study the general properties of amino acids. Okay, so the first property of amino acid is solubility. Now, some amino acids, they are soluble in water, and some are soluble in organic solvents, but they are insoluble in water. So all amino acids, and actually most of them, they are relatively soluble in water, although some of these amino acids, they are sparingly soluble or they are insoluble in water. And their solubility is actually due to the charge side chains or the R groups. Okay, for example, in the case of aspartame, in the last class we had studied the structures of these amino acids. So these are some of these amino acids, they have the charged R group. Okay, aspartate, glutamate, glycine, and arginine. Okay, so these are the examples of the amino acids which are soluble due to their side chain, okay, the R group or the side chain. Now phenylalanine, okay, phenylalanine is the amino acid which is insoluble in water. Okay, it is insoluble in water, but then it is soluble in organic solvent. And we have another amino acid, that is cysteine. Cysteine is also insoluble in water. Okay, it is not soluble in water, and also it is insoluble in dilute mineral acid. And serine and threonine, these two amino acids, they are very much soluble in water. Okay, these two amino acids, they are soluble in water. And the solubility of amino acid is also dependent on the pH of the solvent. Okay, it is dependent on the pH of the solvent, then the ionic strength, the dielectric constant, and the temperature. So these are the four factors. Okay, these are the four factors by which the solubility of amino acid is dependent. Okay, the pH solvent, the ionic strength, dielectric constant, as well as temperature. So these are the four important factors which affect the solubility of the amino acid. That is the pH solvent, the ionic strength, that is the concentration of ions in the solution. Then we have the dielectric constant, that is the amount of the electric polarity energy in a given volume of a solution. Okay, the amount of electric potential. Now the next property we have here is the taste. Okay, the next general property of amino acid is the taste. Now the amino acids, all the amino acids, usually they are either sweet, bitter, or tasteless. Okay, so all the amino acids, some of them, they are either sweet or bitter, or they are tasteless. Now for example, you will see that glycine, then valine, serine, threonine, all these amino acids, these four amino acids, they are usually sweet in taste. And leucine is bitter in taste, and arginine and lysine, they are usually tasteless. Okay, so this is another property of amino acids, and some amino acids, they are used as flavoring agents. Okay, flavoring agents, for example, vanillin. Okay, this is used in ice cream. And we have monosodium glutamate. Okay, glutamate acid. This one, it is also known by the common name Ajinomoto. This one, it is used for flavoring noodles. Okay, it is used as a flavoring agent in noodles. So some of the amino acids, they are sweet, some are bitter, and some are tasteless. Now coming to the next property, that is the melting point. Okay, that is the melting point. So amino acids, they are unique among the organic compounds, okay, to have a high melting point. So they have a very high melting point. And in general, the amino acids, they have a melting point more than 200 degrees centigrade, and some of the amino acids, they even have more than 300 degrees centigrade. So this is a very, very high melting point as compared to other organic compounds. So the amino acids are unique among organic compounds to have a very high melting point. And coming to the next property, okay, the next property here we have Zwitter ions, or also known as dipolar ions. Okay, so here all amino acids, they exist in aqueous solution as a Zwitter ion. Okay, so in solution, just remember that the amino acids, they exist as Zwitter ions, and the Zwitter ion is an ion which contains both a positive as well as a negative charged. Okay, so here just remember that the amino acids, they exist as Zwitter ions. That means they have in solution, the amino acids, they have both positive and negative charge, and that is why the amino acid inside a solution, it can act either as an acid, okay, that is a proton donor. Why? Because it has a positive charge. It can act as an acid, that is a proton donor, or it can act as a base, that is the proton acceptor. Okay, why? Because it has a negative charge, so it can accept one more proton. Okay, so just remember that the amino acids, they have this property inside the solution. They can act as the Zwitter ions. For example, here you can see, suppose this is the amino acid. Here I'm not writing the side chain, I'm just writing as R here, meaning the side chain. It can be anything. Okay, so here we have like this. You see one edge from here, it has come and bind to this amino group. So this is with extra proton, and this is with one less proton, so it is negative. So inside the solution, they exist in this form, okay, the Zwitter ionic form. So this Zwitter ion, it can act both as an acid, a proton donor, and a base, that is a proton acceptor. Now suppose here we have Okay, so you see here this is the carboxylic group COOH, and this is the amino group. So what happened in aqueous solution, when you keep this amino acid inside a solution, this bond, it will come here, and this proton, it will come and bind here. So we will get our CH plus with extra proton, and here we have one less proton. Okay, so it has become negative. So both positive and negative charge are present inside a solution. Okay, this is inside the aqueous solution. Now another important property of amino acid is isoelectric point. Okay, this is another important property of the amino acids. Now when an amino acid is dissolved in acidic solution, okay, it is dissolved in acidic solution, what happened? The amino group, it gets protonated to this one. Okay, this group, it gets protonated to this one, just like in this case. And thus, in acidic solution, the whole amino acid, it gets positively charged, and so it will migrate towards the cathode. Okay, it will migrate towards the cathode. Just remember this, inside the acidic solution, only the amino group, it will get protonated to this one, and thus the whole amino acid, it will become positively charged, so it will get attracted to the cathode, which is negative, right, to the cathode pole. And when an amino acid is dissolved in basic solution, okay, when it is dissolved in basic solution, the COOH group, it will get dissolved to COO minus. So then in this case, the whole amino acid, it will become negatively charged. Okay, it will become negatively charged, and thus in this case, it will migrate towards the anode. And also at certain pH, what happened? The amino acid will carry both the positive and the negative charge. Okay, just like in this case, you've seen that here in this case, the amino acid, it carries both the positive and the negative charge. Now this specific pH where there is no migration of amino acid either towards the cathode or the anode, this particular pH in an electric field is called the isoelectric point. Okay, I hope it's clear. Just remember, this one is inside the aqueous solution. The amino acid, it can act as both, okay, both as proton donor and as proton acceptor, because it carries both the charge. But here it is different, okay. In acidic solution, it can carry only the positive charge, and thus it gets attracted towards the cathode. While in basic solution, the amino acid, it carries only the negative charge, so it gets attracted towards the anode. But at certain pH, okay, at certain pH level, where the amino acid, it can exist as a Zwitter ion, okay, Zwitter ion, both positive and negative charge it can carry. In this case, it will not be attracted either towards the cathode or the anode. That in an electric field, that point is called the isoelectric point. Okay, that field is referred to as the isoelectric point. Now here in case of acidic pH, what happened? You will see, suppose this is the amino acid, right? COOH and NH2. Now in acidic solution, we have an extra proton. Okay, because in acid we know that it is proton donor. Sorry, you see here, if acid meaning proton donor, so there will be a, if the amino acid is present in acidic solution, then the proton will come and bind to this amino acid inside the acidic pH. Okay, the extra proton. So this will get the extra proton and it will become positively charged. You see, this one, it does not go and bind there. Because this is acidic pH, so the extra proton will come from this one, from here, the acidic pH. It will come and bind to the amino group. So that is why the whole amino acid, it becomes positively charged. Alright, so same thing in the case of basic pH. In basic pH, here we will have, it will lose one proton. It will lose one proton, so it will have extra electron. This will be lost. Okay, so it will become COO minus in case of basic pH. So in that case, this will become totally negatively charged and it will be attracted to the anode. So here all the 20 amino acids, they have different isoelectric point. Meaning, for example, here the alanine, okay, alanine, it has the isoelectric point at pH 6. Okay, so this is the isoelectric point of alanine. That because I told you that at certain pH they will become both positive and negatively charged. So in that point they will not be attracted to either positive either cathode or anode. So that pH is referred to as the isoelectric point of any amino acid. So alanine, its isoelectric point is at pH 6. And for example, glutamate, it has the isoelectric point at 5.65. Then lysine, it has isoelectric point at 9.74 and arginine at 10.76. Okay, so we see that all these amino acids, they have different isoelectric point. This is the pH where these amino acids, they will become both positive and negatively charged, and they will not be attracted to any of the cathode or the anode. So the next important property of amino acid is ultraviolet absorption. Ultraviolet absorption. So in this property of amino acids, only 3 amino acids. Okay, remember when we study the classification of amino acids, we have the amino acid which fall under this class, the aromatic amino acids. Okay, so these 3 amino acids, which are also the aromatic amino acids, that is tyrosine, then we have phenylalanine, and also tryptophan. These 3 amino acids, they can absorb UV light. Okay, they have this property of ultraviolet absorption. And tyrosine, it can absorb the ultraviolet light at a wavelength of 278 nanometer. Phenylalanine can absorb at 260 nanometer, and tryptophan, it can absorb UV light at a wavelength of only 280 nanometer. Okay, so these are the 3 amino acids which can absorb ultraviolet light. And the next property of amino acid is the optical property. So out of the 20 amino acids, only glycine, if you remember the structure of glycine, okay, the structure of glycine is very simple, but if you remember, here we have the amino group, then the carboxyl group, the hydrogen, and the R group in case of glycine is only a single hydrogen atom. Okay, so this is the structure of glycine. It is very simple. Since in both the sites here we have hydrogen, hydrogen, so the glycine is, that means it has the symmetric structure. Okay, it is symmetric. Why? Because in both sides, in both these bonds, we have hydrogen atom which are bonded to this carbon atom. Whereas if you remember the structure of all other 19 amino acids and those 2 new amino acids, if you remember that they are all asymmetric. Why? Because you will find that the R group is different, and that means here all the groups which are attached to the carbon atom, to this alpha carbon atom, they are all different. So that is why these amino acids, they are asymmetric. Okay, except glycine, which is symmetric, since it's the side chain here is hydrogen only. Now these amino acids, since they are asymmetric, that means they have the four different groups which are attached to a single carbon atom. If you remember, exactly just like in the case of carbohydrates, this carbon atom is usually referred to as the chiral carbon, remember, because it is attached to 4 different groups. So this alpha carbon here, if you remember, in case of carbohydrate, if they have this carbon which is attached to 4 different groups in their structure, that means they can rotate the plane of polarized light. Okay, remember, they can either rotate the plane polarized light either clockwise, which is indicated by positive, by plus sign, or anti-clockwise, which is indicated by a minus sign. Okay, and in this way, because the amino acid, they has this specific rotation towards, because most of these 20 amino acids, they have specific rotation towards the left. Okay, they have specific rotation towards the left. So most of the amino acids are L-amino acids. They are designated as L-amino acid. They have the L configuration. And the specific rotation of each of these amino acid, it varies with pH. Okay, so it varies with pH. At different pH, they have different rotation of. So now we come to the configuration. Okay, configuration of amino acids. So just like in carbohydrate, if you remember, we have a D and L sugars. Okay, D and L configuration. So in case of amino acids also, since they have this chiral carbon which is attached to 4 different groups, the asymmetric carbon, so they can rotate the plane of polarized light either towards the left or the right, okay, clockwise or anti-clockwise direction. Now most of the amino acid, actually all of the amino acid found in proteins, they are in L configuration. Okay, they are in L configuration. All these standard amino acids which are found in protein. And on isolation, and also when dissolved in water. Okay, when dissolved in water, some of the amino acid, they show L or D configuration. Okay, because you know that in a solution, these amino acid, they can exist either in L configuration or D configuration, and depending on their configuration, where they rotate the plane polarized light towards the left or right. Now on synthesize in the lab, a mixture of both L and D configuration can be formed, and that mixture is referred to as the racemate mixture, okay, which is inactive. Why it is inactive? Because it exists in both L and D configuration, so it is inactive. It cannot rotate the plane of polarized light, and that mixture is racemic mixture. Now, the example of the L and D configuration here, for example, of the trionene, okay, trionene, this is the structure of trionene. This is the carboxylic group, the hydrogen amino group, and this is the side chain or the R group. So if you take a look at this one, this carbon, okay, the hydrogen is on the right and the amino group is on the left. So this configuration is L configuration. Okay, this is trionene molecule. And that is why in D configuration, the trionene can be written this way. Okay, you see the NH2 is on this side and the H group is on this side. So this is D-threonine. This R group, it remains the same. Sorry, this one also, it will become like this. So this is the D configuration and this is the L configuration of trionene amino acid. Okay, this is the configuration, the L and D configuration of the amino acid. Now, suppose here in L configuration, okay, in this L configuration, I have this group. Okay, suppose this one, this side chain is this way. Now you see that in this case both the same group, that is the hydrogen, is on the same side. Okay, so that is why this configuration, it is still the L configuration, but it is referred to as allothreonine. Okay, it is referred to as allothreonine. Why? Because the groups allo here, meaning the groups, they are present on the same side. As you can see here, the hydrogen, they are on the same side. Okay, so this we have the L-threonine, the trionine amino acid in L configuration, but here this is referred to as the allothreonine because the hydrogen atoms, they are on the same side. Okay, with regard to these carbon atoms, they are on the same side, so they are referred to as allothreonine. Same here, if I want to write this as D-allothreonine, then I will keep this one that side. Okay, so that the hydrogen, it will become in this side. Don't change this. Okay, because this is the L-threonine, you can see it remains the same, and here also in D-threonine, this will remain the same. Okay, see, the NH2 group will be on this side and the hydrogen will be on this side. But in case of allothreonine, then the OH will be on the same side. The hydrogens, they are on the same side. Okay, so this is referred to as the D-allothreonine. So just like in the case of carbohydrate sugar molecules, we have D and L configuration. Amino acid also, they exist in two configuration, and most of the standard amino acids, all of the standard amino acids that are present in proteins, they are usually found in this configuration, in L configuration. Okay, for example, here I'm giving the L-threonine. But in lab, on isolation or when dissolved in water, these amino acids, they show the either L or some amino acid will show a D configuration.